Okay, great. Thank you very much for the kind introduction and for the invitation. So as it was mentioned, I'm talking about taking the evolutionary shortcut, kleptoplastidy, and the evolution of plastids. So first, just to give a brief introduction on the evolution of plastids. So this really is the leap that brought photosynthesis, which was previously a bacterial invention, into the eukaryotes. And that this event that had a shared evolutionary origin for all of the algae and the land plants, and therefore led to this greening of Earth, both in terms of on the land, but also of the creating the plankton environments in the oceans. And as I'm sure everyone is aware, it's now widely accepted that the organelles, the mitochondria, and the plastids had an endosymbiotic origin, though this was controversial for a long period of time, but several people really championed and pushed forward this idea, and one of which was Lynn Margulis. But now we accept this, and we call these events primary endosymbioses, and this is because that it's the bacteria that is the symbiont with a eukaryote. So in the case of the plastids, we believe that there was a cyanobacteria symbiont that was engulfed by a protist host, and over a vast amount of time, it slowly became part of a cell, lost its independence, lost a lot of its genes, some to the nucleus, some just lost, and is now just this subcellular structure. This event happened in the ancestor of the Archaeoplastida and led to the three lineages that contain the primary plastids, the glaucophytes, the red algae, and the green algae, with the land plants coming off from the green algae. Now, for these primary plastids, they're surrounded by two membranes, and this primary endosymbiotic event is evolutionarily very rare, having happened just once, except for the exception of Pollinella chromatophora, which we can discuss a bit later if you want. But for the vast majority, it seems that it has just happened once. So it seems a rare event. But what happens more often is then, so that was a primary endosymbiosis, which leads us to this state, that this host can then itself become a symbiont in a second host. So we call this sort of this serial process of endosymbiosis, and this is then a second host and a secondary endosymbiosis. Now, a few lineages get stuck in this intermediate stage, so that the nucleus of the first host is still there, when we call this the nucleomorph. But many cases have actually gone so far that the only sort of real sign that it was a secondary endosymbiont is the number of membranes we see. So if you remember, the primary plastids have these two membranes around them, but if it's a secondary plastid, because we have these extra membranes that got involved with the secondary event, they then are surrounded by 3 or 4 membranes. So this has happened multiple independent times, and it leads to a slightly different membrane structure around these plastids. So it's still being worked out exactly how many times these secondary events have happened, but if this is the tree of life depiction where the primary plastid lineages are here, and you can see that there's thought to be 2 events of secondary endosymbiosis from the green lineages, and it's a little more controversial in the red, but roughly sort of 4, maybe more, to different lineages from the red. But the important point here is this enabled it to really spread out to a very diverse systems of hosts. And then another process that's involved here that I'm not going to speak on, but just to mention as an alternative, is you can also get ongoing secondary endosymbioses. So it's not that it always is this constant reduction process just to the plastids, but you can also have that the whole algae remains as a symbiont. And this is far more common throughout a whole spectrum of lineages. So I just gave 3 examples here. Was a paramecium host that has hundreds of green algae chlorella within it, lichen that are the endosymbiosis between fungi and algal species, and coral that have the dinoflagellate endosymbionts within them. And that's what allows them to photosynthesize. So this is a far more common and really sort of spreads it even further. But even though it doesn't always reduce all the way that we often sort of think about how you would evolve an organelle, here a plastid, is that it's this process of reduction from an endosymbiosis, so that you're getting the gene reduction as it's no longer living in the environment, so can afford to lose some of its functions. In turn, it loses its dependence, its autonomy, especially the cell cycle and cell division will start to be controlled by the host. And eventually, as we start getting towards an organelle stage, protein import becomes the only way that the endosymbiont organelle can survive at this point. And there's two other theories I just want to introduce. So one is the thought here that's been termed the shopping bag hypothesis. And this is the idea that it seems quite difficult how you would think, how does a host have the gene set that it needs to control an endosymbiont? Like, it's not going to have genes just waiting for something that hasn't happened yet. So you need to sort of think about how they could evolve to get these control mechanisms. And one of the ideas is that maybe these pre-adaptations could come from previous interactions with different plastids. And the name comes from the fact that you may be carrying around a shopping bag that has the name of one store on it, but it doesn't mean that all your shopping comes from that store. Like, you're going to have placed things in just because it's a convenient way, so that you can't just look on the outside and expect everything on the inside to be from that same source. So that this would allow them to get those pre-adaptations and therefore really integrate and build this relationship. And the second idea I just want to introduce is called the ratchet model. And this is the idea that there are some key acquisitions that really help bring these interactions together and very tightly knit them. And these are thought to be the metabolic transporters, because of course, if you need to exchange metabolites, which is what these photosynthetic relationships are built on, that it's the carbon fixation that the symbiont is doing. So you need to transport the metabolites, and also if the endosymbiont needs proteins, then the protein import machinery is also very important. So that if these things are achieved, then you suddenly unlock sort of a next level of integration. Suddenly you can exchange metabolites, suddenly you can import proteins. So that these key points can really act as ratchets in the integration. But talking about the main topic of today, which is kleptoplastidy, as this alternative but related form of interaction that you can have. So Rachel said she'd already covered this, but so it comes from klepto, that means thief, and it means stealing plastids from the prey that you eat. And importantly, it allows the host to become transiently photosynthetic, which allows a sort of mixotrophic lifestyle, which is quite an important transition for an organism. Like, ecologically, it opens up an entirely different way of survival, and it allows some sort of fundamental facts. If we were to think that it's the plants that photosynthesize and animals that normally eat the plants, but kleptoplastidy has allowed even some animals to photosynthesize, such as this photogenic sea slug. The plastids, once they're stolen, are termed kleptoplasts, and there is a huge diversity in them. But what the life cycle means is that, so you have a host that needs to feed on its prey. It then sequesters the plastids from them, digesting the rest of the prey and getting rid of them. And in some cases, they can even split them, but they can't divide them. But this sort of happens continuously. And there's a lot of diversity in these relationships. So some last only for a few days, but others can last up to 30 months, for instance, in the Ross Sea dinoflagellate. There's diversity in terms of their specificity. So some are very specialized and will only ever take plastids from one specific prey. Others are generalist and are willing to take plastids from a wide range of sources. There's diversity in terms of their dependence. Some will engage in kleptoplasty depending on the environmental conditions, but there are also obligate hosts that have to engage in kleptoplasty, otherwise they won't survive. And this leads to different levels of integration. And there's also a huge number of species that engage in this. So I just wanted to give a very brief overview, but we have the two animal groups that engage in this, the sea slugs and the flatworms. There's the foraminifera that are protists that produce these fantastic shells, which they can house kleptoplasts in. There is the ciliate group, which is a big group of protists, and the dinoflagellates, which also have many species that engage in kleptoplasty. So just overall, that it's a hugely diverse process, which covers both very specialized and very generalist hosts and happens all over the tree of life. So this again expanded which lineages can have photosynthesis. As we study it more and more, we're realizing that it has a large ecological impact. And it was even recently estimated that about 40% of ciliate biomass is probably kleptoplasts. And when it blooms, some of the species of Mesodynium can become almost the entirety of the biomass in their environment. There are also hypotheses currently trying to predict how much it impacts in ecosystems and nutrient cycles. Now, these are models, so I'm not sure how much the numbers are accurate, but people, when they add these processes into the models, see a large impact. So I think it's important processes to also consider at these bigger scales. And there was one recent prediction that said that if you include kleptoplasty in the model, you can get this big increase in the transfer of organic matter to the larger trophic levels, really drives an increase in carbon storage in the deep ocean. And other recent work has also theorized how it could have a specific role in the formation of the deep chlorophyll maximum, along with the nutrient limitations and light that are also at play there. And what is interesting is also how these differences in how generalist or specialist they are determine their geographical distributions. So work has seen that the generalists really occupy the high nutrient environments, which ends up being fairly coastal. But the specialists look like an intermediate between the generalist and then the photosymbionts, who are the photosymbionts can survive at far more low nutrient areas, but the specialists become these intermediates between them. So it's not just about their lifestyle, but it impacts their distribution and also the ecology that they're interacting with in these environments. But the central crux for kleptoplasty is how are they capable of maintaining their plastids? When plastids only encode roughly 10% of the proteins that they need, where do the rest come from, or how is it possible to delay this procedure? Because when they don't have, so the rest of the 90% is normally provided by the nucleus that they've evolved with. But when you take them away from that nucleus, then the kleptoplastic host doesn't have that co-evolutionary history, so it can't provide them, which is what leads to the eventual degradation of kleptoplasty and therefore the need to refeed eventually. But there are many species that can maintain their kleptoplasts for an awfully long period of time, with over a year in the Rossi dinoflagellate. Also, one of the sea slugs, Elsea chloratica, can maintain for a year, and some of the foraminifera, which seems a hugely long period of time when you don't have that co-evolutionary history. And one point just to mention here is what really speeds up the process of the protein degradation is light. So light both provides the energy in the system, it is the reason that you engage in the relationship, but light is also very dangerous and both leads to the photo damage, particularly of the photo system proteins, but also if there's excessive light, then you get these reactive oxygen species, and they not only damage proteins and DNA themselves, but also inhibit the repair processes. So you can really spiral out of control if you end up in a life environment that you're not adapted to. So in terms of how to deal and maintain kleptoplasts, there are a number of different strategies, which I will give a lightning introduction to. So some of them are based about, therefore, how to avoid highlight, so that you can evolve sort of phototaxis by the host, and therefore when it has kleptoplasts, it will move away from dangerous light. You can shield kleptoplasts, so the sea slugs have been shown to sort of close their wings in a protective way if the light is too high. You can also alter the distribution of kleptoplasts within the cells, so like chloroplasts are rearranged in leaf cells. And these are pictures showing the fluorescence within foraminifera cell structure in different light conditions. Some of them also produce what are called sunscreen molecules, so molecules within the cells that protect from high UV environments. And some, interestingly enough, also just seem to have associate with highly stable plastids. So there's also variation in how stable plastids are to begin with. So that it was found that the kleptoplasts of one of the longest maintaining sea slugs are themselves just incredibly stable. So the isolated plastids just alone from the prey maintain their electron transport and carbon fixation for days. They remain physically intact for 2 weeks. And they have a high number of plastid-encoded genes compared to some of the other species. So it seems that there's also, if you pick the right prey, that you also have an advantage with how much maintenance is required. It's also a theory that you might be able to dual target mitochondria proteins to the kleptoplast, but this hasn't been shown yet in these systems. And then, of course, the final point is that you can just provide the proteins. But of course, if the host is capable of providing the proteins that the kleptoplast needs, then you can maintain them for longer. And within this, there's a few different ways of doing it. A few species, only 2 to date, retain the prey nucleus. So that, of course, it is the prey nucleus that has the proteins encoded on them. So if you hold on to that, then you can still make them. It was first found in Mesodynium rubrum and has been since found in one other dinoflagellate species. And they've seen that the transcriptional profile really changes. So it's not just like a photosymbiosis, where, of course, the nucleus is there as well. The nucleus is separately retained to the plastid. So they're both within their own vacuoles. And the host, the kleptoplastic host, is capable of manipulating the transcriptional profile of the nucleus. So it's a lot of manipulation that goes involved with it. But in one sense, it just changes the question, because then the kleptoplasty only lasts as long as the prey nucleus lasts, and then it breaks down as well. So you sort of just, you solve one problem, but you also create a new problem. So the nucleuses aren't easy to maintain either. There's also some sense that you don't need to maintain the whole nucleus. Maybe some of them are capable of just maintaining transcripts or just smaller pieces of DNA. But then we have found a few examples where there really are the host encoded targeted proteins, and they have been capable of really integrating these proteins into their genomes. And that's what I'm going to spend the rest of today talking about. But briefly, just to say, therefore, that we can split these strategies in two, that some of them are about avoiding the need for the renewal of proteins, and then we have others that are saying, okay, we'll just provide the proteins. So host-encoded targeted proteins have been identified in 4 kleptoplastic systems so far. It was first found in a dinoflagellate host called Dinophysis, which is obligate and lasts for a few months without refeeding. In this Ross C dinoflagellate, which is the one that has the longest ability to maintain its kleptoplasts, which is also obligate, they found 15 targeted proteins. In a different, so Rapaza, which is a euglenozoa, it's also obligate. It lasts for 30 days, so a bit shorter than the others, but they estimated a huge number of targeted proteins within the system. And then the one that worked on with Rachel, which is Moringa. And we identified that it had 14 targeted proteins there. So I'm going to talk through how we found these within the Moringa system. So Moringa is a pico-eukaryote that is globally distributed, and when it blooms can be highly abundant, but was very understudied for a long period of time. It has these distinctive silica spines and scales around it. And it was consistently observed to have these green or golden bodies within it, but when you looked, had what appeared to be chlorophyll fluorescence, which was why it was historically classed as an algal. So they thought it was just an algal that could photosynthesize. But then recent work by Vasily was looking at samples taken off the west coast of Sweden. So they're uncultured, so all the work has to be done from environmental samples. And by sequencing the 18S region, reclassified it as a centrohelid. Now, what this means is centrohelids are this group that have no known plastids. So they're not expected to have a secondary event. So there'd be no known reason why there would be plastids in this system. We continued working with these environmental samples, and we took 15 single cells taken from the environment. We used a process of amplification, which allows you to get enough genomic material from a single cell to then sequence it, so that you really get the sequence of one cell as opposed to a sort of a population, which is how it's normally done. And using these, we could again get this 18S, this identifier region. And what we couldn't see was a secondary nucleus of an endosymbiont, so that it's not that photosymbiosis that we talked about briefly, where the whole cell is maintained, but it appeared to just be the plastids. Then when we looked at this 18S, what we saw was just from this phylogenetic tree, and just the take home here is just that we identified these two different groups, so that there seemed to be some diversity even within this system. And then when we looked on the symbiont side of things, the plastids, we identified their identifiable region, which is the 16S, and they grouped from within the Stromenopals, which is a group that are known to have a secondary endosymbiont from the red lineage. And within these, which was interesting, is we saw the two groups, but which matched the host group. So we have what we call group 1 and group 2. But here they don't group together like they do in the host, but rather you can see that there are free living, the green hair species in between them, which is potentially shows that there was sort of an independent origin within these two groups. But then what we wanted to start asking is, are they permanent or kleptoplasts? And we designed qPCR primers and probes that are very targeted approach, and we continued using these environmental samples and could look for just our probes matched either the host identifiable region or one of the plastid genes here, the RBCL. And then we could look at samples from across a year and see when they were there and when they weren't there. So here the top row is the host, and we see that the purple, which is group 2, and green is group 1. So we see that the group 2 hosts are there throughout the whole year. So they're just stably there. Their abundance changes, but they were found there the whole time. But group 1 is only there for a few months of the year. And the plastids were also not found throughout the whole year. Group 2 is again found for more months than group 1, but there are months where for roughly half a year, we couldn't find the targeted sequence that we looked for from these plastids. So then the question was, what's happening in these months? Are they losing their plastids completely, or are they switching? And this is when our sequences came from. And this, just the losing in itself also suggests kleptoplasty. But what's happening in these months? And in these months, we did some cardfish sampling, which allowed us to make this probe. So this green signal matches only Moringa cells. And from here, we could see that the chlorophyll fluorescence was present in these months where we didn't have the qPCR, and matched the light microscopy distributions that we saw. So this made us think that there probably was plastids throughout the whole time, but there was likely switching. And then for the final point, to test the integration, we created more robust groups. So we brought all our samples together into 3 main groups. And from here, we did a very targeted search for 41 known host-encoded plastid-associated proteins and for 35 transporters. And what was really important when we were looking for them was to look not just for the genes, but also their content. So to really make sure that they were encoded within the host genome, you have to look therefore at their neighboring genes. And we also manually checked for misassemblies and predicted whether there would be targeting. So not just that it's in the genome, but is it targeted to then be moved to the kleptoplast? And how these results are shown, just briefly, is that there's little boxes like this, and the bigger colors either side show the sort of the origin, so whether it's in the host genome. So it's blue if we really found a strong signal of it being in the host genome. If we predict targeting, then it's the color of the small square, which is green if it's plastid targeting. And then the color of the central one is the protein origin, which comes from a diverse number of sources. So these results, there's a lot of detail in this. I don't really expect you, we'll talk through some of it in a bit, so I don't expect you to take it all through, but at this point, it's just noticing that we did find and predict a lot of kleptoplast targeted proteins, that we saw them in the 3 different groups that we looked at, that we saw them across a range of functions, and that we saw quite a lot of kleptoplast targeted targeting. So to summarize this, we found 22 candidates across the groups and across functions, 14 of which have this predicted targeting signal. The RNA is mostly from dinoflagellates, which is a completely different group, so would represent horizontal gene transfer, and also the stromanophiles, and the symbont, the dictocophycia, come from within the stromanophile group, so those could potentially be endosymbiontic gene transfer. And there's one example of retargeting of a host protein. All the other genes come from outside sources, but there's one example where it seems to be potentially a duplication event that gained targeting. To focus in on a few examples, that we believe there was potentially cases of there being complementation between the host and the kleptoplast. So here with the iron sulfur cluster biosynthesis, what is normal in dictocophycia is that these 2 genes, the SUFB and SUFC, are both encoded on the plastid. But in our plastids, the SUFB has been lost, and we see 2 cases of it being gained and targeted in the nucleus. So it seems to complement what was lost in the plastid. This was also seen for one of the potential elements of protein import, TIC20, which we saw in one of the group 2, and the group 1 still maintained a plastid copy in the genome. So there's potential mix of the nucleus acquiring things that the plastid has lost. And the last point is also that we found 3 examples of targeted transporters, one of which modifies iron distribution and has been found to be involved in the oxidative stress response in Arabidopsis. One is a sodium-potassium cotransporter, and one of which is a probable sugar transporter, which is obviously an exciting point given that it would provide a way for the photosynthate to move back. So overall, it was a flying introduction to Moringa sphaeria, and our working hypothesis is that Moringa sphaeria seems to show a very selective kleptoplasty, so it only chooses from these dictocophycia. And even within the 2 host groups, they seem to choose very specifically one type. It seems to have seasonal dynamics, that these seem to reappear annually. But despite the fact that it's potentially switches to a different plastid for part of a year, we still identified these candidates for host-encoded plastid genes, which seems to come from a mix of horizontal gene transfer and endosymbiotic gene transfer. This work hasn't shown whether these candidates are functional, so that they're just working on the genome sequence, so we can see that they have this targeting region, but whether the protein actually gets to the kleptoplast and whether it's functional there would still need to be shown. But what it shows is that Moringa sphaeria, compared to the 3 other systems that I mentioned that also had these systems, these proteins, is a more dynamic kleptoplasty. The others are very stable and completely obligate and don't seem to switch. And it's important that it's in a centrohelid because they're not ancestrally plastid bearing. So you're managing to evolve these proteins in a host that has never had a plastid. Whereas you can imagine, so I mentioned that 2 of the others were dinoflagellates, and there they used to have plastids. So then there'll be some of their sort of ancient plastid proteins that they can reuse, which makes it easier. So it also supports both the ratchet model that I mentioned at the beginning, because out of the examples we saw, we did see some for the protein channels, VACTIC20, and also for the metabolic transporters. So already at this stage in a kleptoplasty, so not getting before even the plastid is permanent, you can start to have these systems that really increase the integration. And it would also relate to the shopping bag hypothesis, because you're acquiring these pre-adaptations, these proteins that can be targeted to the kleptoplast and help with integration before the plastid is permanent. But in the case of kleptoplasty, it's also, you do need to extend this to include horizontal gene transfer from prey. And it's also a good example. It now appears that we have these 4 systems, that kleptoplasty having these proteins does not just seem to be a one-off rare example, but seems to be a strategy that has developed independently in multiple different lineages. And it's likely that as we explore the diversity of these systems more, we'll find more examples of these. And so just one more slide. I just wanted to sort of bring it out again, and these could be potential discussion points. So we have these 4 systems with the kleptoplast targeted proteins, and they've been termed evolutionary tipping points, some of these kleptoplasties that are so integrated that they have levels at which we thought would only happen in organelles / the very most stable endosymbioses. And therefore, the question becomes, could a kleptoplasty evolve into a permanent plastid? Or is it always this sort of weird extra thing that happens on the side? Or is it something that actually, when we could be used to just shortcut, and instead of going through this long process of reduction and loss of dependency, you could just steal essentially a plastid that's already been made for you by someone else? So it offers a potential for this shortcut, but this is still very much something that's being considered and discussed in the field. And of course, you would need the building blocks there to be already. So you need to have prey, and you need to have the horizontal gene transfer to gain these proteins potentially from other systems, which means it would only work for a secondary plastid. It's never going to be a process that would allow you to get a primary plastid, but it could be a process that would allow you to get a secondary plastid if the building blocks are there around you. And therefore, questions such as how would you even recognize this? Like, how would you tell if there was a permanent plastid in the system? Would we even be able to tell if it came from a kleptoplasty or from the slow reduction of a secondary endosymbont in the system? So these are very much just completely open questions, and maybe we can discuss some of them now. So just to thank the Moringa Spheria work was done with Rachel and Fabian Berku while at Stockholm and Uppsala University, and was really helped by Irina, Vasily, and Anne, who I forgot to mention her name, but she helped a lot with the analysis. And this is the funding bodies and the sampling unit, the SMHI, that helped us get the samples. And just briefly, I am, however, currently at Heinrich Heine and working with Eva Noack. And thank you very much for listening. Maybe I'll go back to this slide.